afternoon, colleagues and friends. So wonderful to be here in person and to introduce our speaker today, Dr. Oliver Eickelberg. Dr. Eickelberg is a good friend of many people in this room, and it's very gratifying to be able to welcome you to today's Grand Rounds. Just for a quick intro for those of you who aren't familiar with his background, after completing medical school in Lübeck, Dr. Eickelberg completed a postdoc in Basel and then came to Yale, in fact, for 4 years, where he worked in pathology and physiology with Michael Kashgarian and Gerhard Giebisch. He then left Yale, went back to Germany, and started his career at Justus Liebig University in Gießen. And it was there where he was the founding director of the International Graduate College Molecular Biology of the Lung. After succeeding there and discovering a lot of things, he then moved to Munich, where he ascended to the full professorship at Ludwig Maximilians. I can't read my writing there, but you were also the founding chairperson of the Comprehensive Pneumonology Center, and that was a really wonderful thing that you did. And you know, built a center, built this destination for pulmonary research, to the point where in 2016, he was recruited to cross the pond and go to the University of Colorado, where he became professor and section chief. Then moved eastward to University of Pittsburgh, where he currently resides and holds the Simmons Chair for Pulmonary Research, the Vice Chair for Basic Translational Science, and the Executive Vice Chair for Academic Affairs. Dr. Eickelberg has numerous professional memberships, just to name a few: European Respiratory Society, American Thoracic Society, and one that we don't often hear here, the Alexander von Humboldt Association of America. Also the recipient of numerous honors, such as the European Respiratory Society Cellular and Molecular Biology Young Scientist Award, the Gay-Lussac Humboldt Award from the French Ministry of Science, and induction into ASCI in 2018. Dr. Eickelberg has made seminal insights into the field of pulmonary fibrosis, as well as numerous other chronic lung diseases, but it seems to be pulmonary fibrosis where he's making his mark. He's very widely published with first or last authored papers in such esteemed journals as JCI, Aging Cell, Blue Journal, and ERJ, as well as team science publications in high caliber journals such as Cell and Nature. He's been cited almost 35,000 times and has an H index of 98, which is pretty good. I think looking at Dr. Eickelberg's CV and all of his accomplishments, his NIH funding, his publications, what actually shines through all this is a visionary and creative scientist. You can see it back, what, 20, 25 years ago, when you're doing your postdoc here, getting really that fundamental knowledge that's required for a physician scientist to be able to apply the scientific method to the study of biomedicine. And that, that's becoming increasingly rare, but you can see it through the high quality science. It's sort of like no matter what the project is, or no matter what the science is, he's able to follow it because he really has that training. I think that's also reflected by the paradigm shifting work that he's done, and the rigor and reproducibility of all his studies, because this is how we're actually going to shift paradigm and cure fibrosis. But the last thing I'd like to say shines through from this body of work is what the dedicated mentorship and he's a great colleague and leader who elevates everyone around him. And I think we are very fortunate to hear from him and learn from him today. So let's welcome him.
Wow. That's impossible to follow up on. Thanks so much, Erica.
Okay, you can just end the talk now.
Kindness of introduction, and I thank you very much for having me and any questions. Thank you for inviting me here, Naftali, Erica. And it's always, as you've heard, it's always a pleasure to come back to Yale. I can tell you all, Fitkin Auditorium looks exactly the same with the exception of the screens than 25 years ago, but certainly that door is still exactly the same door. And I was also telling earlier that in the coffee place, just walking down the hall, that's where I sat when 9/11 happened. And Didi, a fellow in pediatric pulmonary, myself, Julie, also a fairly pediatric pulmonary. We sat there having coffee and we watched live when the second plane hit the towers. And that was also a memory I never forget, as probably most of you. If we know one place where we were at one time, it was 9/11 when the planes hit the World Trade Center. I think all of us exactly remember where we were. And that was, at that time, this was crazy for Yale because we didn't know how many people would come down from New York at the time, and nobody knew, you know, you have to clear the hospital, you have to make space for everybody what would come down from New York and Manhattan particularly. And we all thought that was the most divisive time, what happened after 9/11, right? Politically. I mean, you all remember, like, you're with us or you're against us. That was the Bush mantra, Bush Jr. mantra. The good old days, I would say. So it's a pleasure to be back in Yale, and I love you, Eben. What can I say? It certainly was a large part of setting me on the road to success. And Erica, you probably mentioned all of the things, and once you sit back and think of where you are and what you did, we're very blessed in what we do. We usually work in beautiful and wonderful communities with very talented people. So here's my disclosure slide. I wouldn't be able to stand here and talk to you about anything scientific if it weren't for all of these people that are here on the slide. So that's the current lab. I've highlighted some of the people that did most or are doing most of the work that I'm going to talk about today. And that's really Marta. And I think Marta was giving a pulmonary research talk like not that long ago. So you probably heard her talk about a pretty similar topic not that long ago. Adriana Yandong, who are research faculty in the lab. Then obviously Mel and I share not only life, but also work partially. So we do run or jointly run a lab and have enjoyed many successes together. Difficult times, easy times, you know, you all know what I'm talking about. And then our beautiful collaborators that we've had in the places Erica has mentioned across the world. I would say the most fun job I currently have is being editor-in-chief for JCI Insight. You all know the ACI. Probably many of you have read JCI Insight issues. Probably many of you have read JCI issues. I want to highlight right here, because this is a community of fibrosis researchers, that we will have this call for submissions on cross-organ fibrosis. So if you have anything in the field of fibrosis, the call is going to go live probably pretty soon. Melanie is one of the issue editors. We do want to have a large representation of organs represented in that issue. So please, if you have any work, let us know, submit it. If you need any inquiries, Melanie's in the auditorium, or send an email to myself. But these are usually issues that are very highly cited, so these topical issues. So if you have good work, if you think you have something ready to be published like early next year, please let us know. Also, I want to highlight that the wonderful thing of being an editor is you can have certain initiatives and you don't need to ask for permission for many things. That's something very nice that I truly enjoy. And we all know that in the coming years, physician scientist careers are not going to be easier. Let's put it like that. There's going to be some difficult times ahead of all of us in terms of what we do, in terms of maintaining our mission at academic medical centers. I do think that physician scientists are going to be very vulnerable, particularly in that early stage of F to T to K career transitions. So we will have dedicated sections at JCI Insight twice a year, usually the April issue before the joint meeting, and then the October issue at the end of the year, where we feature dedicated articles about physician scientists' career, being able to sustain a career, being able to highlight how can we help our early career transitioning scientists or physician scientists. And that's going to be a large emphasis of JCI Insight. And for our first issue, the fun part is what do we do on the cover? And we said, well, why don't we get like all our like young budding scientists, you know, preschool and whatever together and ask them, hey, how do you envision physician scientists? So we had the entire board. I mean, in Pittsburgh, most of our faculty asked, hey, if you have a kid, let them, draw physician scientists. What do they think? So this is going to be a cover issue here. Obviously, this is son of a political person. Highly biased, but still a beautiful representation. Up there, also probably person that Jordan Skizzi's son. Then that's a surgeon, a donor of a surgeon on the upper left, so obviously the scissors. And then on the lower left, a kidney person, and I think you can imagine that's it. Yeah. So, but it was so fun to have the kids, see, hey, how do you envision physician scientists? Just draw something. And these were like age 4 to 8. So this is very early on. And we had a really bunch of submissions to this. You will see them on our social media channels. So once the issue is out, we'll feature every picture individually with the story behind it and so forth. So make sure you go to the social media. But that was such a fun exercise. And the amazing stuff that we got from the kids is really, that's heartwarming in these times. And I think that's the message that we want to send also to our early career physician scientists, that we have your back, at least at ASCI and the journals. It's not going to be an easy ride in the next couple of years, but we try to have you back as much as we can. So on to lungs. I'm going to talk about lung transplantation for the most part of today's talk, which is sort of, I got into it, as Erica mentioned, by investigating tissue fibrosis, originally from the interstitial side of things, but then really moved into chronic rejection after lung transplantation, because that is a very specific compartmentalized fibrosis, as you've seen in a second. And in Pittsburgh, we're very fortunate that we have a large transplant program. So every once in a while, we get, both lungs out of a donor that's not ready to be transplanted, doesn't fulfill transplant criteria. So we get the 2 entire lungs, like with almost no ischemic time. As you know, if you get organs anywhere from the country, you have ischemic times, like somewhere between 6 to 24 hours. So these are like 5 minutes ischemia time. Then they're hooked up to an ex vivo lung perfusion system, and then we can investigate those. That's really a treasure, and that's sort of our gateway also to personalized therapy and localized drug delivery, because we can aerosolize these lungs, we can ventilate the lungs, we can also perfuse the lungs and sort of assess on-target efficacy in a human system, which is pretty rare to do. One paper is just an acceptance that will come out soon. So I want to talk about these unique challenges of lung transplantation. To me, it's almost like the last or the final frontier. It's not that we actually solved a lot in respiratory medicine, solved a lot of diseases, but most diseases end up in lung transplantation, right? Most advanced lung disease ends up in a patient being transplanted. Unfortunately, that is the last remedy for many patients, but we also know that that comes with its own price, because 50% of patients that are transplanted will have some form of chronic rejection after 5 years. I talk about the preclinical animal models in this area, compare some of the animal models, and then focus in on B and plasma cells. And the cell that we really investigate scientifically for the past 10 years, that's a long-lived plasma cell of the lung. And I hope at the end you can understand a little bit why we're so obsessed with these cells. So survival after lung transplantation, difficult to say, but it's again one of these areas in medicine where we haven't moved the needle a bit in the past decades. So here you have a classical survival curve from different decades. This is from the International Society of Lung Transplant. And you see it really hasn't changed over the decades. Still, the 50% survival is somewhere between 5 and 6 years. So there's really been no major therapeutic innovation in being able to address that chronic rejection period. Obviously, you see the acute rejection period with immunosuppression certainly has helped over some of the past decades, but the long-term outcomes are still the same from whenever we started lung transplant on. And that's sort of a bit the challenge that we dive into. If you ask, well, what are these life-limiting factors after lung transplantation, that's indicated on the left graph here, you see that as you move longer after the time of transplantation, chronic lung allograft dysfunction, CLAD in yellow here, which is chronic rejection, is really the major factor that limits life in post-transplant patients. I'm not going to talk too much about this period of primary graft dysfunction up to 72 hours after transplant, which is not our focus of research, or the acute rejection periods that still patients are subject to, but can be handled sometimes better than the chronic rejection periods. So why do I say this sort of reflects a little bit tissue fibrosis, just in a different compartment than what we're used to in interstitial lung fibrosis? So on the upper left part, you have a histological picture from one of the early New England papers that describe bronchiolitis obliterans, which is one aspect of chronic lung alveolar dysfunction. And you see the fibrosis is really sort of localized around the bronchial structures. You'll see that in real histologies in a bit, but as opposed to interstitial fibrosis, where the fibrosis is localized to these interstitial streaks in the lung, these are really very localized to the bronchial structures. And you have a healthy human lung on the bottom, on the left side, and then a boss lung on the right side. And you see this massive, constrictive obliterans phenotype that sort of gets rid of a normal bronchial structure that happens in most of the patients. Now, despite some of the knowledges that have occurred over the past couple of years in CLAP, still there is no evidence-based guideline how to treat it, and very minimal publications regarding effective treatment regimen for chronic lung alveolar dysfunction. So that's up to us to change in the next couple of years. I think it partially has to do with the fact that it's a very difficult disease to replicate in organisms that we can subject to rigorous scientific research. So many of you will work in some sort of lung disease and you do an animal model, either in a mouse, in a rat, or a pig, or any other animal model that sort of you can test therapeutic efficacy. But lung transplantation is a very difficult model because you have to orthotopically transplant a mouse lung into a recipient mouse, and that surgery is not done easily. So today, you probably have a handful of centers in the US that transplant more than 50 orthotopic mouse lungs a year. We have much more clinical centers that do this on a higher volume, but there's really only a handful of centers that do regular mouse orthotopic lung transplant. Pittsburgh, fortunately, being one of them, which is one of the reasons why we moved to Pittsburgh. The last NIH statement, and who knows if the NIH will do consensus statements in the future, yeah. That will be something we'll have to see. But the last statement was published in a pretty good journal there, JCI Insight, about 10 years ago now. So there's also a bunch of new science that will have to be addressed in the next review. Suffice to say that some of the models on the right that I've indicated here that you find in the literature, but when we started, our aim was, can we generate a model that reflects all the changes that we see in patients after lung transplantation? And I'll take you through some of the data we generated with that model. So here's the team after we moved to Pittsburgh. So Jing Wan is the surgeon that does that trains all of the surgeons now in lung transplant. He was recruited from WashU about 10 years ago. Marta Bueno is basically the PI in the lab now with me. She's in the middle, and then Byron Shannon was our lab manager at the time who headed all of our animal colonies. So in the first year we moved to Pittsburgh, we finished the 127 lung transplant right there. So that team has really been up to speed with also having significant numbers of mouse lung transplants. Because that is a limiting factor. It's not only difficult to do the surgery, but your group sizes are way different than any of your bleomycin groups or whatever, because a surgeon, a very well-trained surgeon, can do 2 lung transplants a day, maybe 3, but that's very exhausting. So there are your group sizes of identical regimens. So it's like 2 to 3, which you then have to replicate in independent cohorts. The surgery is exactly, or most exactly like what you would do with patients, just it's a single lung transplant. It's a single lung that you do in mice. You don't do combined heart and lung. You don't do both lungs. You do one lung. And then if you have the right model, as you see in a sec, you will replicate most of the findings in that model that you see from patients. So here's, that was our wish list when we started the model, and that was started by Natalia, as you see in a sec, about more than 10 years ago. So one is we wanted very reproducible airway remodeling. That's almost the hallmark, or that's where the name is derived from, bronchiolitis obliterans. So parenchymal diffuse fibrosis, we did not want, but we want this localized peri-bronchial fibrosis. We do want a model that has pleural thickening because that is an underlooked phenomenon in most transplant and IPF or any sort of interstitial lung disease for that matter. We wanted to have that selective loss of club cells, which is so well known from patient populations. And then, to be clinically relevant, we also want a functional parameter to mimic our patient population, that usually is a loss or lung function decline. We came up with a variety of standard models and tested those early on and introduced one new model. And the new model is the HLA-2B6. So as mentioned, you take one lung from a donor, you transplant one lung in situ in this exact same orthotopic place into a recipient. If you do a B6 to B6, obviously there's no antigenic stimulus. It's the same, it's a basic syngeneic. There's not going to be any pathology. So every surgery that we do, you always do one sort of HLA-B6 F1 to B6 or mismatch, and at the same time you do, of course, the B6 to B6, so that you basically have your control population at the same. And remember, 2 a day, so you do 1, 1, 1, and so forth. So in our 10-year experience, we never saw any phenotype change when you do syngeneic, so B6 to B6. There really is nothing other than the cuff, where you suture basically the tracheas and the veins together. The HLA to B6 is the one we have now used for most of the part, which is now being rolled out, and many investigators use the HLA to B6 now. And the other classical ones are complete mismatch scenarios, F1 to B6, DBA, DBA, F1 to DBA. Forget the names. The thing is, those are sometimes completely mismatched. Like in the F1 to DBA, you see that's not a lung anymore. So how about that selective loss of club cells? So you see all models investigated in parallel. And I just highlight the HLA to B6 because that is a single antigen in the HLA-B6, in the HLA locus that is inserted into the mouse strain. So that lung that goes from an HLA strain to a B6 strain has one single mismatch from the human genome into the mouse genome, so one single mismatch. That leads to that massive phenotype that you see in a sec. You see here, B6 to B6, HLA-B6, massive loss of club cells, as in the staining indicated here, the loss of green color. You see the quantification of CCSB positive cells on the right, and you can appreciate that overall is a loss in many models. It varies with respect to the effect or size, but you do see the HLA-2B6 has a very robust effect. Lung function is shown here, so we usually to the full Galois of lung function. What I'm showing you here is change in FEV and respiratory capacity. And you do see one thing. Those of you who deal with other mouse models like bleomycin models, for example, these groups are so tight with respect to lung function. That's why we sort of can get away with group sizes of 4 to 6 in these models. If you do 4 for bleomycin, those of you who do it, you know, you can shoot it out of the cage for some of them, and that screws your entire data set. So these are really tight models, particularly with complex assessments such as lung function. So this is sort of where we stand right now, which is still way behind on other areas in medicine. What are some of the remaining questions that, as you see as I go through the talk, is I think age is something that we have not addressed fully, because clinically we know if you transplant an aged lung into a younger person or a young lung into an aged person, you do have different outcomes. There's very good clinical data for it. That has not been investigated well. Size mismatch is another thing, so that's very difficult to address in mouse studies. So why did we focus on and transplant on this plasma cell population I want to talk about? So Natalia really started the whole model, and that's more than 10 years ago, way back in Munich now. She found that this model replicates this selective club cell loss, as shown here. That's in a JCI Insight paper a couple of years ago. So we knew that this was sort of a model that we wanted to take further for mechanistic investigation. When we moved to Denver, then that was the time when single cell analysis, 2008, came a little bit more available. If you remember, those were the times where at least the assays were available. Computational analysis was the bottleneck at the time, still is sometimes today. But you could basically at many core facilities run those assays. So that was the first experiment Natalia then did. Run a single cell analysis of a mismatch versus a control, so HLA to B6 versus a B6 to B6. And you see on the lower right side, those are the cell populations that were changed. So obviously, several cell populations changed, pretty much like in a clinical specimen from a patient undergoing lung transplantation. But the one that caught our eye is that plasma cell population that I circled here with the blue lining, because that was robustly increased in the first analysis we did and every other analysis we since did. So we wanted to look at that plasma cell population, and some of the genes that were changed are shown in the lower right, MCB1 being one of the genes. So if you then look on the cellular level, this is how it looks histology-wise. On the left, B6 to B6 versus HLA to B6. You can see in the macroscopic picture that you have these peribronchial fibrotic patches, right? You can almost see the bronchi shining out of the regular H&E stain here. But if you stain for the cells, NCB1 in the middle in green, you see there's this massive accumulation of cells in the rejected lungs, and the quantification is shown on the right. And now the amazing thing is they always show up in this fibrotic areas. And again, in bronchiolitis obliterans, it's around the airways. It's not in the interstitium like in the pleo or any interstitial lung disease model. Shown here in much more detail, also for healthy and horse lungs, so both in the human and the mouse scenario, this lined up very well. I'm going to show you this histology here, where you can really appreciate that in green you have MCB1 staining. So those are your MCB1 positive plasma cells. And you don't need to see anything else, but you know where the bronchi are, right? They're beautifully outlined bronchial structures. And it's every patient we ever looked at. There's no exception. So it's a really amazingly robust finding. So these cells, which we sort of like in the interstitial meeting that we had earlier today, already showed you some of the data, but these cells are characterized by this pattern, CD138 positive, CD127, MZ1 positive. So these are plasma cells that are tissue resident. That's basically the population of cells we're looking at. They are completely independent spatially from tertiary lymphoid structures. So they do not like emerge from tertiary lymphoid structures. They are in the interstitium. They are around the bronchi in bronchiolitis obliterans, independent of any other structure other than bronchi. And you see they do stain for IgA and IgD as in the stainings below. Since we had that staining, we also asked the question, where if we have a mouse that is sort of deficient in Ig secretion, there is this one mouse, the ADA-C mouse, the ADA mouse that's indicated here, that has a partial deficiency in Ig secretion. So we used that in the transplant model, and that was partially protected. So immunoglobulin secretion, which MZ1 is supposed to be doing, plays some role in pathogenesis, but it doesn't completely prevent the phenotype from bronchiolitis obliterans. So we had to drill more into the sequence of events that would lead to the emergence of that MZ1 positive population and some of the factors that would likely drive that differentiation profile. So Natalia looked more in detail into those B to plasma cell clusters that's indicated on this, and we have the velocity analysis, cytotrace analysis here. Suffice it to say that from all of the analysis we ever did, there seemed to be a trajectory from B1 B cells into that plasma cell cluster. That population is highly MCB1 positive, and there were some transcription factors, as you see in this, that were also pretty highly expressed, highly and specifically expressed in that cluster. And that's, for example, the DHAG41 shown here, which is one of the transcription factors which we're currently modifying, which would prevent that cluster of cells to emerge. And by blocking that transcription factor, we can actually prevent bronchiolitis obliterans in mice. We don't know if we can reverse it at this time, because that, as I tell you in a sec, is a whole set of new conundrum and mouse problems to be addressed. But at this point, again, suffice it to say, this population of cells can actually differentiate and secrete immunoglobulins. So you have actually on the left, ex vivo immunoglobulin secretion of these sorted B cell subpopulations. And you see if you sort them from the mismatch, so from the chronic rejected lung, they have higher amounts of Ig secretion than if you sort them from the regular syngeneic B6 to B6. And on the right side, you can also see if you do immunoglobulin isotyping, it's mostly an IgG to C phenotype. That's what's mostly increased in these lungs that are rejected, but also IgM and IgA. Next question we ask is then, does this really recapitulate in patients? Because very little was known about subtypes in this specimen. And you see here on this graph, so this is from a cohort of both patients and respective controls from South Florida, Tampa General Hospital. And this from Nirmal Sharma, who's now actually at Harvard, so this was his cohort at the time. And you see that indeed, pretty much like in the mouse, there are some Ig subtypes which are highly upregulated in that rejected population. Now this is lavage levels now, and some immunoglobulins which are unchanged. So it is indeed a very specific process that's occurring in these antigen-producing cell types. But that's where really, I was like, I never wanted to be an immunologist. I hated immunology. I mean, I was at Yale during the hate times of immunology. You still have hate times of immunology here. But I mean, I never wanted to go into cell differentiation kinetics of B cells, but here we are. Okay. So this plasma cell we're talking about is likely derived via this differentiation cycle. And there are certain mouse models that we've looked at that affect the differentiation of or the emergence of this plasma cell population. So the MUMT mice have no B cells at all, they're protected. So that was a very early study of Natalia's. That was her first animal experiment she did. And I showed you the Eickelberg knockout mice, which are partially deficient in antibody secretion. They are partially protected. They will block B to plasma cell differentiation at these points indicated here. There are two other mouse strains that will block several other differentiation pathways, right? CD19. And again, we're dealing with two different mouse models. We're always doing with the knockout model or the cell-specific knockout, but then also with the diphtheria toxin receptor model in order to deplete the cells once we establish fibrosis. So the question would be, can you prevent disease from occurring, but can you also reverse disease once it is established? So we always need these two models. So a gazillion of mouse colonies and a gazillion of, unfortunately, costs associated with that. And then we also generated these mice, and all mice are now finally breeding. But it took the better part of 2 years to generate these mice. So the one that's very interesting to us is this DHLG41 knockout. I mentioned to you that's one of the transcription factors that is activated during that terminal or B1B, B1A to plasma cell differentiation. So that mouse is protected actually in everything we ever tested it now for, like both bleomycin as well as chronic rejection. And then we have the MZV1 knockout, the inducible MZV1, and the MZV1J chain knockouts to really unequivocally get a handle on, okay, can you reverse disease once it's established? And can you pinpoint which mechanism it really is? Is it the MZV1 protein? Is it the emergence of the plasma cell? Or is it the antibody secretory phenotype? So this is where we are right now. If you use the MZV1 knockout, you can prevent the cell type from emerging. That's shown here. So on the left, you have the HLA to the MZV1 knockout mouse. You see there's no emergence of that plasma cell population, and the quantification you see on the right. You can see the same basically in lung function as mentioned earlier and on histology here, that you reduce the phenotype or you reduce the chronic rejection phenotype from emerging. And again, that's assessing all of these parameters that I mentioned previously. So at this point, we're sort of at, I mean, we are pretty confident that we have a cell type that is involved in tissue fibrosis. In this case, chronic rejection after lung transplant, but I'll show you in a second also in other fibrotic diseases. Every sample we ever looked at from any sort of fibrosis has accumulation of these cell types. So I've never seen fibrosis without this cell type. So that is an exciting therapeutic area right now because these cells, you don't find them in normal tissue in relevant numbers. So the off-target effect of any sort of compound that would affect longevity or existence of the cell type would likely have very little off-target effects, side effects, that is. So that's currently where we are. When it comes to how do we move the needle in transplant or chronic rejection research in general, I think our challenge right now is that we need to extend the observation times for these post-surgical outcomes, same as with our patients, right? So 50% of clot occurs 5 years after transplant. How do we model that in mice? Does it get worse? Does it resolve after time? Can we accelerate somehow decline? I would say right now we use two model systems for all intervention approaches, so two different scenarios of chronic rejection. I think integrating clinically relevant outcomes like lung function to me is essential in all of these models. And then it's this question of age. How does a young lung in an old recipient fare versus an old lung in young recipients? Can you break tolerance like either way easier or not? So those are like exciting scientific questions. We're beginning to watch phenotypes up to 20 weeks right now. Now this is just histology, but we'll gather like function data. So that'll be an interesting aspect for progression or not of the model. Ideally, it would be progressive. Ideally, you could do survival studies. I don't know at this time. But I show you why we are so obsessed with the cell type. So I hope I convinced you that they sort of play a role in chronic rejection. Anybody not convinced?
Have you used the tuxedo?
So, interesting question. The question was if we use rituximab, so CD20 depletion or CD20 mediated depletion of B cells, which is used in a lot of like clinical scenarios. So that's CD20 depletion is not effective systemic on these cells because if you run it, these cells still emerge. There's been so we've done it, but there's been very good published data on renal transplant in human studies where the plasma cell population is not affected by circulating CD20 cells, which again makes the whole thing complicated for us too, because we believe this is a tissue originating plasma cell. It's not from a circulating pool. Everything, when we have this transplant scenario, we can do color matching, right? So we can transplant, green cells into red mice and so forth. They're not emerging from the circulation. So that is unfortunately a complicated issue for this, because that would be wonderful to have. So then, yeah.
Since you offered the opportunity,
I did.
I'm not one to ever pass up that up. So, you've shown an association at this point, but there's a mechanism question here, which is obviously, to me, is what's the antigen? What are they generating these IgG against? Because I think if that would really help understand, if there's a true connection to the development of fibrosis, or whether this is at this point, a biomarker of the process.
Yeah, so the most unsatisfying momentum of this talk is I can't give you a mechanism. And that's as unsatisfying to me as to you and everybody else in the room. And the challenge, I outlined the challenge in a couple of slides. We do have a lot of evidence for mechanisms that I go into, but I don't have an unequivocal mechanism. And this is why we have not submitted the work yet, and we're still looking for it, but we don't have a definitive mechanism. It is likely that there's many stories around in multiple organ systems emerging that B plasma cells are in effect a negative regulator of epithelial healing via actually mediator release and intercalating into these niches that restore the epithelial layer. And I think that is the mechanism, but I can't prove it as of today. So the experiments are going. I think that's where it's heading. I don't think it's the Ig secretion entirely. I think it's a mediator release and a niche enabling activity of that plasma cell population, or a niche disabling activity if they emerge. I can't prove that to you as of today.
But I mean, B cells can present antigens. There are antigen presenters also, and they're also effectors. So why not just the regular B cell function? They are in the place, particularly if they're close to a follicle. They're presenting the antigen. They're also effectors.
So these cells are never close to a tertiary lymphoid follicle. They're never close to a classical follicle. They're always distant from follicles. I mean, actually, you see in some of the stains also.
You see them in mediastinal lymph nodes, or
That's another conundrum. There's very little MCB1 staining in mediastinal in hilar lymph nodes that we resect from the same ILD or chronic rejected lungs. And we think this is not the same population because we have done all these in vitro differentiation steps from the hilar lymph nodes. And I don't know if I think I had those in the talk earlier, but then we ran out of time. That's not the same population. I'm convinced of it by now. And I think the the single cell data proves that we currently have. But that's also the difficulty because we cannot isolate the cells from the lung and investigate them functionally. You have to in vitro differentiate them because you can't get them out of the matrix and you can't make them live ex tissue. But we also can't fully differentiate them into the cell type that we see currently. So, I mean, that is a little bit the challenge of this. I mentioned I never wanted to be an immunologist because of precisely that.
They're functioning like a tertiary follicle. They're functioning.
But they don't emerge from follicles. They are not spatially in proximity of follicles.
So I think we've had a really good discussion, and let's let our speaker get back to his talk.
All right, I will do. So let's look at idiopathic pulmonary fibrosis for a second. So as mentioned, this is not a peribronchial fibrosis, interstitial fibrosis. So we published like way back then with Herbert in Munich, 2017, it was in the Blue Journal, this very small cohort where we did proteomic analysis. And MCB1, as shown there, was already one of the most robust, most highly upregulated proteins in interstitial lung fibrosis at the time. So this is really ILD patients. It was one of the best proteins that was equally regulated on RNA and protein. Because as you know, in the lung, RNA and protein are not always equivalently found, but some of them, as shown here, are very expressed both in protein and RNA level. So MCB1 was one of these top. And third, it was one of the top proteins that we found in interstitial lung fibrosis and scleroderma-associated lesions in skin. So if you look at two different body compartments, skin and lung, on the right side, there you see MCB1, equally upregulated both. So by now, we're fully convinced that MCB1 is a common regulator of any fibrotic process that we ever looked at. And I've extended that to liver now, to skin, to kidney, and to lung, multiple forms in the lung, as you saw. So we believe this is like one of the true profibrotic, non-organ specific mediators of fibrosis. And really, this became substantiated when we ran this proteome analysis, which I've introduced earlier today. So this is 60 samples of 60 ILD, 60 scleroderma ILD, 60 COPD, 60 control, and then 30 POS. And I cut this short due to interest of time. If you look at the classical volcano plots, this is what you see, pretty similar for scleroderma ILD and IPF. If you look at the heat maps, you see some specific patterns, some not so specific patterns, and some that would indicate cross-organ fibrosis mediators. MCB1 is really one of the most cross-organ mediators that we ever found thus far. This is from an analysis with systems immunology. Jishnu Das at the University of Pittsburgh runs these programs that identifies these essential nodes. And I've introduced that earlier. MCB1 is one of these essential nodes that seems to be a latent factor for disease pathogenesis, meaning it's causally pathogenesis, and it's likely one of the key nodes factors. So this is an analysis we done completely independent of the entire first half of my talk. So knowing what we had in terms of mechanistic data and then seeing that in an unbiased analysis, this shows up at one of the central nodes of tissue fibrosis, of course, to us was super comforting. So we thought like, okay, here we have all the functional data, clearly plays a role. Now we have an basically sort of hypothesis-free analysis using a pretty significant data set. And again, the same protein shows up. We thought this is really something that plays a role in fibrosis. So here's just the bleomycin model and human ILD samples. And again, you see the same thing I showed you earlier for post-transplant CLAD. You see on the left side in the middle there, this is the abundance of the MCB1 positive plasma cell population. And it skyrockets, for example, in scleroderma ILD. Even compared to the BOS samples I've shown you, if you look at a scleroderma ILD lung, these cells are skyrocketing. They are so abundant in these lungs. In the bleomycin model on the right side, you see very isolated cells emerge in the single bleomycin model, but in the repetitive model, which is closer to what's happening in ILD, you see emergence of that cell type. So likely the cell type needs time to differentiate, evolve, and then sort of be present in the tissues. Also in the bleo model, however, the mice are protected from fibrosis when you have when you don't have MCB1 in the lungs. And just to emphasize this, here's an ILD lung. So this is a scleroderma ILD lung in red. This is what you just asked earlier, right? So in red, you have the MCB1 staining, so these are all the plasma cells. In green, you have follicles, CD20 staining, so B cell follicles. And there's no spatial localization or emergence in any lung we've ever looked at. So there seems to be an absolute independence of the differentiation pathway from the classical differentiation kinetics we know from follicles to B cells. And I close with this data slide here. We've since now looked in fibrotic liver disease. The nice thing is, we're sort of limited in the lung, right? Because we biopsy in ILD patients, maybe if we're lucky once for diagnosis, but that's it. In transplant, we do surveillance biopsies, so we do biopsies 6 months, 12 months, so you get tissue in a longitudinal intra-individual fashion. But the liver folks, they biopsy the heck out of their patients. You get like biopsies like galore, and they have markers of disease progression which are found in the serum. So that gives us a real nice avenue of trying to identify circulating biomarkers that would reflect the abundance of the cell type in the liver. This is hard to do in the lung. So since we've hooked up with our Pittsburgh Liver Center folks, here you see healthy liver center on the right, and then MASH. And again, they change their nomenclature daily, so that may be outdated already. But you see in red is a smooth muscle actin stain, so that is the vessels or the SMA layers. And in green, you see the MZ1 positive cell population. And again, MASH, like in interstitial lung disease, these cells are everywhere. If you have highly fibrotic liver or viral hepatitis, for example, you find these cells in every sample we ever looked at. And as I mentioned, the nice thing is you can do sort of staging of samples and then measure abundance of the cell types in these biopsies. So this is done here. And you see on the lower right, they really nicely emerge with stage of fibrosis. So that the higher stage fibrosis, the more cells these patients have. I leave you with what is MCB1, because I talked about the whole cell biology and molecular biology of MCB1. It's an intracellular folding enzyme, basically. It's a very small, soluble enzyme. It's of very limited structure. It belongs to this sapid family, supposedly like proteins. It is believed, but not ultimately proven, that it helps in the folding of proteins that undergo secretory pathway secretion. So that is likely the function. But again, that's the most assigned function that you'll find in PubMed and most of the people that have investigated MCB1. There's not a whole ton of literature on the protein. So where are we with it? I'm pretty confident that we do have a global marker of fibrosis. That I do believe you can tell stages of fibrosis by investigating MZ1 positive populations. I do believe they're absolutely essential for the emergence of fibrosis. I don't know if this is indeed a population of cells or a protein of interest that would be, if therapeutically targeted, be able to reverse fibrosis, but I certainly hope it would be. And again, I think the mechanism at the end of the day, I didn't show you all of the in vitro differentiation data, is likely restoring that niche, which is prevented to heal in the presence of these plasma cells. Because one thing that's clear is wherever you have fibrosis, you have these cells. And fibrosis is always connected to these cells, and we can't get it out of that fibrotic tissue. So they must be cemented into the tissue and likely have a lot of cell-to-cell contacts, which sort of are formed with the epithelial mesenchymal niche. This is where we are. We want these targeted with plasma cells markers to easier get a handle of them. We want to obviously characterize the population, which is ongoing. Ask the question, if we deplete them, is that therapeutically relevant? And then hopefully identify a surrogate peripheral marker that would give you a sense of staging of fibrosis or MZ1 load. And with this, thanks a lot for listening and for having me.
Great. So we've already had a really good extensive discussion, but we fortunately have time for more. I'll go with Rob first, since I've known him longer, and then we can go to Mayor.
There's a microphone right there.
The customer was mentioned, but obviously the plasma drug that myeloma folks use.
We test. so we're testing those in vitro right now and see if they prevent the emergence of that MZ1 positive plasma cell. Because during the in vitro differentiation, at least, we have two different plasma cell phenotypes. One is highly MZ1 expressed, one is not. So we're currently doing these tests with, sort of bortezomib and all of these inhibitors out there and see if we can slew the differentiation pathway. And if there would be an avenue for clinical trial.
Great. Dr. Sauer?
All right. You get preferential treatment there and getting that back.
It's okay. I'll always defer to Rob. So beautiful talk. And also just especially for fellows, like, right, how exquisite those models are that you're doing. Like, it's really incredible. My question is really about the plasma cells. So you mentioned multiple times they're not from tertiary lymphoid structures, and yet plasma cells shouldn't be dividing in tissue. And so these are probably differentiating from another cell population. And so is there some sort of local proliferation somewhere that is independent of the plasma cells? And do you know where the source is?
So I don't. What we internally discuss is, you guys have worked on macrophages, right? Remember when the innate macrophage population emerged, right? From the yolk sac macrophages, people didn't believe it at all that would be another differentiation pathway. So I sort of look at that for analogy. Again, I'm no immunologist by training, but it seems to me there is an alternative differentiation pathway that's operative only in a fibrotic tissue that will drive the expansion of the cell type, because for sure it's not the classical differentiation pathway. We do know they are senescent, many of them. If that triggers a senescent associated phenotype, that would drive fibrosis. There's good evidence that could happen. But in the absence of having a robust characterization in vitro, right? Like, and then functional analysis. So giving flow cultures, giving it to PCLS of normal versus fibrotic, right? I get hesitant to call it a day with a definitive idea, but that's where we currently are. Unfortunately, it's a tricky population of cells.
This is really fascinating, and it sort of brings me back to some work from a guy we know very well, Marvin Schwartz, which described
deposits. Yes,
exactly.
Yes.
So the IPF lung does have immune complexes, and for years we've ignored it. And on the other hand, at least our work has not shown that B cells or regular B cells are abnormal in this disease, at least not in IPF. So the question, one is maybe their mechanistic role is still in secreting IgG, and especially in places they're not supposed to be. Right. So that's one question. The other question, I really loved your comment about senescence, because as I was listening to you, I was thinking, okay, our aberrant basal cells are very similar. We don't know where they come from. We cannot really culture them. And we can mimic their features, but we can actually cannot replicate them. So the question is, maybe it is the unifying hypothesis would be senescence in those very few cells that do end up replicating in the lung in response to injury. And maybe these are the SASP cells in the lung.
So we have our opening editorial for the crossover fibrosis issue, Dr. Königshoff, right there. This is the theory. I love it. I think that is under-investigated. The challenge that we have in the field, as you mentioned, right, is in the absence of being able to culture these cells and get a defined mechanistic approach to it, it's very hard to convince like a non-fibrosis person of the mechanistic value of it, right? So I think that's both with the aberrant epithelial cells as well as the plasma. That's the challenge right now. I think it's a challenge even for the fibroblasts because we're not culturing the right cells there. I think with respect to the, it was so funny, right? Our first multidisciplinary ILD conference in Colorado, Marvin still was part of them. He's not anymore, but he was part of them. And that's the first time I read his New England paper, because he said, did you read my New England paper from, 1945 or whatever it was? And indeed, he described these Ig deposits in IPF specimen, and they've sort of flown under the radar, right? Over the past decades, but they are there. Every time we stain for IGs in the IPF lung, these cells light up like crazy. So And I think it's part of the mechanism, right? The IDA mouse experiment shows that. I don't think it's the only but it's certainly part of it. Yeah.
Great. So we have time for one last question from Dr. Takyar.
It's, I mean, they all say it's amazing. I think it will take a little time for us to digest and come up with real thing, but two questions, I mean, two issues. One of them is, to a real B-cell biologist, this is a one station in the B-cell maturation, right? B1 to the plasma cell. Now, why is that the stop? Has anybody asked or any answer? Like, what's happening to T cell? Because they are presenting antigen, which antigen you may find by sequencing T cells to see which antigen it is. So that's the more complicated one. The simple one is, I'm sure you're going to ask, so what happens to immunity? Do they survive well? Like, what happens with an infection? You're taking out the plasma cells if you target.
Yeah. So as of now, we can tell the difference in infection, but we haven't run, but that's like it. And actually it's a dirty facility. So that sort of comforts me, right? Because the transplant is done outside of our, clean facilities. It's in like sort of a transit facility, which is a bit dirty. And then the post-transplant surveillance fields is done there. They're not going back to the clean facilities once they're out for transplant. There's no sign of any increased infection in the post-transplant mice that have the MTB1 knockout. With respect to the B cell function, B2 plasma, so I mentioned Jishnu Das, who's the computational biologist. The other person we're collaborating with in the scleroderma B50 is Sarinder Singh. He's a hardcore B cell biologist. So he's sort of asking the same question where he doesn't have an answer to. How do you get a handle on those differentiation kinetics in the absence of a classical follicle scenario, right? And I think that's what we're faced with. So when we do the higher lymph nodes, we can't get the same percentage of cells differentiating, but we can't isolate the cells from the lungs because it's so hard to get. So Maur's point to the tertiary lymphoid follicles in COPD or even in ILD, I mean, we see them everywhere. Can we recreate that like somehow in vitro? So Cambes is doing this bone marrow on a chip technology with us. So I don't know if that would help in getting a more or a better handle on the populations. I mean, lots of exciting avenues right now, if there just were funding for it. I have to close on a positive.
Yeah, I think that's a great way to end. So thanks again for a wonderful presentation, and to our audience,